Hello, Hey everyone. My name is Charlie Ahrens, and I am currently a graduate student at the University of Connecticut Health Center. And I'm doing my research on GSD in Dr. Yomuk Lee's lab here at UConn Health. And so today I'm just going to be presenting some of our more recent findings on glycogen storage disease type 9. So just a brief reminder that it's extremely important that the body tightly regulates the way it creates and stores energy. And this process balances your blood sugar levels. And so, for example, if you've just eaten a big meal, your blood sugar levels increase, stimulating your body to convert glucose into glycogen to store. And this process is called, over here on the right, glycogenesis. And then on the flip side, when you haven't eaten for a while, your blood sugar levels reduce, stimulating your body to break down that glycogen into glucose through the process called glycogenolysis. And this, again, will raise your glucose levels back up to a normal range. And so two very key components in this glycogenolysis process are called glycogen phosphorylase and phosphorylase kinase. So just a little bit of a closer look into these enzymes. So glycogen phosphorylase, which is actually the key enzyme in GSD type 6, is typically in an inactive form until it is phosphorylated by its partner, this phosphorylase kinase, where it converts into an active form and then can break down glycogen into a molecule called glucose 1-phosphate, and then it can carry on through the process until it's converted into glucose. So as you can see here, the phosphorylase kinase, which is the key enzyme when talking about GSD type 9, is comprised of 4 subunits, and previous studies and animal models have looked at type gamma and alpha. However, very little has looked at the type or the beta subunit. So with our investigations, we wanted to look at what happens when you have a deficiency in this beta subunit. What happens with the energy metabolism. And so there are some key features that we know about GSD type 9 beta just from clinical studies. However, there's still a lot more that we can learn from an animal model such as a mouse model. And so coming into this, we wanted to evaluate the underlying mechanism of the typically more mild phenotypes seen in GSD type 9 beta. We wanted to explore how prolonged fasting could potentially lead to liver complications. As well as with this mouse model, we wanted to evaluate the disease progression as they matured. And so we utilized a mouse model harboring a PHK beta subunit deficiency, and it was created using a gene editing technology called CRISPR-Cas9. And so the exon 4 of the PHKB gene was cut out, rendering the whole gene dysfunctional. And so we could genotype our mice using PCR, seen on the left here. And then on the right, we confirmed that we had a complete knockout of this PHKB gene using our gene expression analysis. And so we see here that it was fully knocked out. And so initially, we really wanted to assess and see if our mouse model recapitulates the phenotypes seen in human patients. And so one of the main phenotypes is hepatomegaly, or the enlarged liver. And you can see here that our mice did indeed have larger livers compared to normal wild type mice. And then to quantify this, we took the weight of the liver as a percent of the total body weight. So here you can see that the normal wild type mice liver weight percent is around 5%, whereas our knockout mice with the enlarged liver is upwards of around 7.5 to 8%. And additionally, we know from clinical studies that patients typically have lower blood sugar levels through fasting periods. So we check that with our mice as well. And we can see here that although the knockout mice were not in a hypoglycemic range, they did consistently have sustained lower blood glucose levels throughout all fasting time periods, as well as throughout the fasting, we see a drastic increase in blood ketones. And so these ketones are produced during the breakdown of fats, which typically happens when stored sugars aren't available. And so these findings, along with the liver size findings, show that our mouse model does recapitulate what we know about the human phenotype. And so we really wanted to investigate what's attributing to this phenotype with the lower blood glucose level, but still able to maintain throughout a fasting period. And so we looked at different energy metabolism processes, and the two main ones here are glycogenolysis and gluconeogenesis. And so the main differences between these two processes are that glycogenolysis gives you glucose from the breakdown of glycogen, whereas gluconeogenesis produces glucose from non-carbohydrate substrates such as pyruvate or lactate. And so to first analyze glycogenolysis, we looked at both the hepatic glycogen and hepatic free glucose in our model. And seen here is also, we also kind of looked at it with a previously created and known model for GSD type 6. And you can see that the type 6 are in the gray bars here. They have high levels of hepatic glycogen and very low levels of hepatic free glucose, and those levels really do not change throughout the fasting. On the other side, with the type 9 in red, we do again see the increased level of hepatic glycogen, so higher storage of glycogen. However, through a fasting period, these levels do actually decrease, which corresponds with a seen increase in hepatic free glucose throughout a fasting period, which sort of gave us the idea that there may be some partial glycogen breakdown in GSD type 9 beta. And so to kind of look at this further, we looked at the actual glycogen phosphorylase activity. So again, we can see here in gray, the type 6 mice have very little to no glycogen phosphorylase activity, which would be expected because that's the gene that they're deficient in. However, interestingly, we do see a significant increase compared to the type 6. In our type 9, we see a significant increase in the phosphorylase activity, although it is greatly reduced from a normal wild type mouse. There is some activity there, so leading us to say that there's partial glycogenolytic activity in GSD type 9 beta. So we also wanted to look at some of the alternative pathways. And so one of those, again, being the gluconeogenesis, which is creating glucose from non-carbohydrate substrates, such as pyruvate or lactate. And so to evaluate this, we used pyruvate tolerance test, which you just inject a small amount of pyruvate into the mouse. And so here we can see that after the injection, the knockout mice had a significant increase of blood glucose levels after the injection compared to the wild type, suggesting that they may be more sensitive to the pyruvate and that gluconeogenesis might be playing a larger role in these mice. And additionally, we checked the gene expression of some of the key genes in gluconeogenesis, and some of our results, such as FBP1, also back up our findings that gluconeogenesis could be upregulated in these mice, helping kind of sustain that blood glucose level through fasting. And additionally, a third process for sort of energy metabolism is lipogenesis and lipid degradation. And so here are some of the key genes in lipogenesis, lipid degradation, or sort of the transcription factors related to these processes. And although none of them, although there aren't really any significant changes in the wild type or in the knockout compared to the wild type, we do see that the knockout are more volatile. The gene expression is more volatile, suggesting that there could potentially be some upregulation of this method for energy metabolism. And additionally, we know from some clinical case studies that liver fibrosis have been seen in some type 9 patients. And so we wanted to look at the profibrogenic gene expression in our type 9 beta mouse model. And overall, the gene expressions of profibrogenic genes were not very significant. Was not a very significant change from the wild type to the knockout. However, there was a couple such as CTGF, which is connective tissue growth factor, and Quol1A1, which is sort of lays out the plan for collagen in tissues, did have a significant increase at certain time points in our mice. However, it wasn't sustained through the fasting period, which sort of suggests it might be very minimal. And here's just a bigger list of the other profibrogenic genes that we looked at. So again, there's very minimal change in these gene expressions. And lastly, we were able to take a look at the liver histology, the histology of the cells of the liver. So the first panel here, we can see that we're looking at the morphology of the cells, and we can see that this bottom panel of the knockouts has the cells are greatly increased. So there's much larger cells in the liver, and this is largely due to the glycogen buildup in the cells of the knockout mice, which we can see in the second staining. So all of these darker purple areas in this bottom panel of the knockouts are the buildup of glycogen. And you can see much more accumulation than in the wild type. Next, we took a quick look at a trichrome staining, which is staining for collagen. So in our knockouts, we do see some minimal collagen buildup in this dark bluish purple color of the staining, but again, it's minimal. And lastly, we looked at an oil red staining, which in this figure, the red little dots are actually lipid particles. And so we can see that, interestingly, the knockouts have a lot less fats or lipid particles accumulating in these cells of the liver. And so overall, from this, we found that this model does mimic the human disorder with the hepatomegaly, the lower fasting blood glucose levels, as well as the increased ketone levels during fasting. We interestingly found that they're capable of sustaining energy homeostasis during fasting through a couple of different methods. We found that they have partial glycogenolytic activity, an increase in gluconeogenesis, and potentially an upregulation of fatty acid oxidation. As well as we found very minimal profibrogenic features in the liver of our type 9 beta mice. And with that, I would just like to thank Dr. Lee and all of the current and previous lab members that helped with this project, as well as all of you for tuning in. So thank you.